Experimental designs. In a true experimental design, subjects are randomly assigned to at least two comparison groups. An experiment is theoretically able to exert control over most threats to internal validity, providing the strongest evidence for causal relationships. The randomized control trial is considered the gold standard of true experimental designs. Experimental designs may be differentiated according to how subjects are assigned to groups in completely randomized designs, also referred to as between-subject designs. Subjects are assigned to independent groups using a randomization procedure. In a randomized block design, subjects are first classified according to an attribute or blocking variable, for example, gender or age, and then randomized to treatment groups. A design in which subjects act as their own control is called a within-subject design or a repeated measure design. The pre-test post-test control group design is the basic structure of a randomized control trial. It is used to compare two or more groups that are formed by random assignment. One group receives the experimental variable and the other acts as the control. These independent groups are also called treatment arms of the study. Both groups are tested prior to and following treatment. The groups differ solely on the basis of what occurs between measurements. Therefore, changes from pre-test to post-test that appear in the experimental group but not the control can be reasonably attributed to the intervention. There are several variations in this basic design. There can be more than one experimental group. Another design is referred to as a post-test only control group design. A factorial design incorporates two or more independent variables with independent groups of subjects randomly assigned to various combinations of levels of the two variables. Although such designs can theoretically be expanded to include any number of variables, clinical studies usually involve two or three at the most. Factorial designs are described according to their dimensions or number of factors, so that a two-way or two-factor design has two independent variables. A three-way design has three independent variables. In this example, the two independent variables are intensity of exercise, A, and location of exercise, B, each with two levels, 2 times 2. One group, A1, B1, will engage in moderate exercise at home. A second group, A2, B1, will engage in vigorous exercise at home. A third group, A1, B2, will engage in moderate exercise at a community center. And the fourth group, A2, B2, will engage in vigorous exercise at a community center. The two independent variables are completely crossed in this design, which means that every level of one factor is represented at every level of the other factor. Each of the four groups represents a unique combination of the levels of these variables. This design allows us to ask three questions of the data. 1. Is there a differential effect of moderate versus vigorous exercise? 2. Is there a differential effect of exercising at home or at a community center? And 3. What is the interaction between intensity and location of exercise? The answers to the first two questions are obtained by examining the main effect of each independent variable, with scores collapsed across the second independent variable. This means that we can look at the overall effect of intensity of exercise without taking into account any differential effect of location. Therefore, we would have 30 subjects representing each intensity. The main effect of location is also analyzed without differentiating intensity. Each main effect is essentially a single-factor experiment. The third question addresses the interaction effect between the two independent variables. This question represents the essential difference between single-factor and multi-factor experiments. Interaction occurs when the effect of one variable varies at different levels of the second variable. For example, we might find that moderate exercise intensity is more effective in changing exercise behavior, but only when performed at a community center. There are many research questions for which control can be substantially increased by using a repeated measures design, where one group of subjects is tested under all conditions and each subject acts as his own control. Conceptually, a repeated measures design can be considered a series of trials, each with a single subject. Therefore, such a design is also called a within subjects design because treatment effects are associated with differences observed within a subject across treatment conditions rather than between subjects across randomized groups. The major advantage of this design is the ability to control for the potential influence of individual differences. It is a fairly safe assumption that important subject characteristics such as age, sex, motivation, and intelligence will remain constant throughout the course of an experiment. One disadvantage of this design is the potential for practice effects, or the learning effect that can take place when one individual repeats a task over and over. Another disadvantage is the potential for carryover effects when one subject is exposed to multiple treatment conditions. Because subjects are exposed to multiple treatment conditions in a repeated measures design, there must be some concern about the potentially biasing effect of test sequence. That is, the researcher must determine if responses might be dependent on which condition preceded which other condition. One solution to the problem of order effects is to randomize the order of presentation of each subject, often by the flip of a coin, so that there is no bias involved in choosing the order of testing. Another solution uses a Latin square, which is a matrix composed of equal numbers of rows and columns, designating random permutations of sequence combinations. When only two levels of independent variable are repeated, a preferred method to control for order effects is to counterbalance the treatment conditions so that their order is systematically varied. This creates a crossover design in which half the subjects receive treatment A followed by B, and half receive B followed by A. This design is especially useful when treatment conditions are immediately reversible. When the treatment has some cumulative effect, however, a washout period is essential, allowing a common baseline for each treatment condition. The washout period must be long enough to eliminate any prolonged effects of the treatment. Quasi-experimental designs utilize similar structures to experimental designs but will lack either random assignment or comparison groups, or both. They often involve non-equivalent groups that may differ from each other in many ways in addition to differences between treatment conditions. Therefore, the degree of control is reduced. There are several design study types associated with quasi-experimental. For example, there is a one-group pre-test, post-test design that involves one set of repeated measurements taken before and after treatment on one group of subjects. The effect of treatment is determined by measuring the difference between pre-test and post-test scores. The independent variable is time with two levels, pre-test and post-test. Treatment is not an independent variable, but all subjects receive the intervention. Single subject designs provide an alternative approach that allows us to draw conclusions about the effects of treatment based on the responses of a single patient under controlled conditions. This design involves the systematic collection of repeated measurements of a behavioral response over time, usually at frequent, regular intervals. The second core element of a single subject design is the delineation of at least two testing periods, or phases: a baseline phase, prior to treatment, and an intervention phase. The target behavior is measured repeatedly across both baseline and intervention phases. The baseline phase provides information about responses during a period of no treatment or a control condition. Design phases are traditionally plotted on a line graph with magnitude of the target behavior along the y-axis and time along the x-axis. Using conventional notation, the baseline period is represented by the letter A and the intervention period by the letter B. To facilitate description, this design, with one baseline phase and one intervention phase, is called the A-B design. Single subject design is a repeated measure design. Think about the application of this study design in terms of internal and external validity. Two characteristics of baseline data are important for interpretation of clinical outcomes: stability, which reflects the consistency of response over time, and trend or slope, which shows the rate of change in behavior. The most desirable baseline pattern demonstrates a constant level of behavior with minimal variability, indicating that the target behavior is not changing. Figure 12.2a therefore represents changes that are observed after the intervention is introduced, can be confidently attributed to a treatment effect. A variable baseline, figure 12.2b, can present a problem for interpretation. When this type of pattern emerges, it is generally advisable to continue to collect baseline data until some stability is achieved. An accelerating baseline, figure 12.2c, or decelerating baseline, figure 12.2d, indicates that a change in the target behavior is occurring without intervention. Either type or trend may represent improvement or deterioration depending on the target behavior. The ABA design represents one baseline following the intervention. The premise of this design lies in its ability to show that behavioral changes are evident only in the presence of the intervention during Phase B. Experimental control and clinical relevance can be strengthened through the use of an AB design, which provides initial baseline data and ends on an intervention phase. The major advantage of this design is that it provides two opportunities to evaluate the effects of the intervention. In an ABCA design, A is baseline, B is treatment 1, and C is treatment 2. When practical replication cannot be achieved, a multiple baseline design can be used, where effects are replicated across subjects, across treatment conditions, or across multiple target behaviors. There are several design types in your textbook. At this point, you just need to know that they exist. This graph demonstrates that improvement is noted only with the application of the treatment. There are a few ways to interpret data using a single subject design data. There is a visual analysis, essentially evaluating stability and trend lines in a graph. There are procedures to calculate these types of lines. This graph represents the two standard deviation band method. The mean and standard deviation is calculated for a data set. The mean is plotted on a graph along with two standard deviations, one above the mean and one below the mean. If two consecutive data points fall outside the standard deviation line, statistical significance is considered. A third method, the C-statistic, estimates trends in time series data. The special appeal of single-subject research is that it focuses on clinical outcomes and can provide data for clinical decision-making. To demonstrate external validity, it is necessary to replicate the results of single-subject experiments across several subjects, or by repeating the study on the same subject. Systematic replication is used to demonstrate that findings can be observed under conditions different from those encountered in the initial experiment. Treatment plans are not developed around isolated patient problems, nor are they based on partitioned treatment components. In the long run, then, the true applicability of research data will depend on our ability to demonstrate that treatment packages can be successfully applied to patients with multiple behaviors that tend to cluster together. This is known as clinical replication. Such a procedure may be considered field testing, that is, clinical replication becomes one with actual practice. Clinical replication is an advanced procedure, which can occur only after requirements for direct and systematic replication have been satisfied. There are articles posted for class. If you prefer a hard copy, please bring it to class. It is strongly suggested that you read the articles once before class to familiarize yourself with content, since you will take a closer look at the content together in class. You will compare and contrast experimental design studies.